Okay, good afternoon and welcome. My name is Ryan Tappy and I work with the ALS Association in our Mid-Atlantic Territory. Thank you for joining us for this ALS Association webinar. There are a few things we want you to know before we begin. There will be time at the end of this webinar for questions. You should see on your screens an option for Q&A. You can type questions you have there at any time and we will review them at the end of the webinar to provide answers. You can also submit your questions in our chat box feature. We do have a very short survey that we are asking that everyone complete after the webinar. The survey will appear in your browser once the webinar ends, but it will also be emailed to everyone who attends today. This survey helps us know your thoughts on the webinar and topics you would like us to consider in the future. The past few months, we have been working to highlight various ALS research and clinical trials that are being conducted in the territory and across the country. We continue that trend today with a presentation on 2 ALS research studies being conducted out of the Pittsburgh area. Our speaker today is Dr. Christi Kolarcik. Dr. Kolarcik is a research assistant professor in the Department of Pathology at the University of Pittsburgh School of Medicine. After completing undergraduate work in biology and chemistry at the Schreier Honors College of the Pennsylvania State University, Dr. Kolarcik received her PhD with honors in cellular and molecular pathology. She continued her fellowships at the University of Pittsburgh, first in the Swanson School of Engineering and the Department of Bioengineering, and then in the School of Medicine. Her research team is currently involved with multiple projects that focus on furthering the understanding of basic biological mechanisms underlying ALS and neural circuit dysfunction, as well as developing therapeutic technology and strategies. She is a member of our National ALS Association's Board of Trustees and has been devoted to working with the ALS community for over 15 years. So, without further ado, I will hand it over to Dr. Kolarcik for the presentation.
Thank you so much, Ryan, and it's really a pleasure to be with everyone today. What I have here are a couple images of the research buildings at the University of Pittsburgh. Since we are at a time of year where it's dark most of the time, I've included images for us in the evening. And this is the building in which my lab is located, and it's probably right about here if you were to peek in the window there. So. Before I get started on a discussion of these research studies, I wanted to give you guys a little bit more about me to give you a sense of my connection and deep passion and dedication for the ALS community. So I started my graduate work in July of 2005, and then I attended my first ALS event in August of 2005. So pretty close together in the time frame there. I attended a Walk to Defeat ALS at one of the parks around Pittsburgh. And then after that first walk, I volunteered to be on the walk planning committee for the subsequent year. This just catalyzed my involvement with the ALS community. I was first involved locally with the Western Pennsylvania chapter. And then, as Ryan mentioned, I was on the National Board of Trustees. I just recently rolled off of that position, but still work on the research committee at the national level. I've been involved in a lot of different efforts over the years, including making assistive technologies a priority in the ALS Association. And this was connected to the work that I did after my graduate studies. And what you can see from some of these pictures here is that I've been involved in a lot of advocacy efforts as well. Hopefully you can see my little pointer. This is a beautiful group that we had from Philadelphia and Pittsburgh, another group here. And then when the pandemic threw us some curveballs, we also did some advocacy online as well. And I just wanted to share with everyone that's on today that when I first started working in this field, people would ask if I had a personal connection, if someone in my family was affected by ALS. And if you would have asked me initially, the answer was no. I didn't really know anyone with ALS. And so what I'd like for you to know now is that my involvement over these years has really led to a very different answer now. So it is only personal for me now. So just wanted to share a little bit of that with you guys before we kind of got into some of the research presentation. So you have a little bit of a better sense of where I'm coming from. And you can also see, you can probably, or at least I can date some of these pictures by which kids are in them and at what age they are. So you can see that this really is our family's mission is to try to leave ALS a little bit better than we found it. So as Ryan sort of alluded to, I have worn different hats and I continue to wear those different hats. I am at the University of Pittsburgh now in the School of Medicine. My home department is the Department of Pathology. And these are the current positions that I hold within the ALS Association. So again, vice chair of the research committee, and I also serve on the Western Pennsylvania Leadership Council. And I also do some consulting work for things related to ALS through GuidePoint, the consulting firm. But again, all of these things are really in my personal and professional effort to create a world without ALS or to improve the world for people affected by ALS. And so in terms of what my laboratory's focus is, one of our overarching research questions is really, why are certain motor neurons vulnerable to the disease process? What makes them affected by the disease while others are seemingly unaffected? And so I just provide this one slide to give you an overview of the fact that our work involves studies in animal models, studies in cell culture-based models, and then research that involves human volunteer research subjects. So that's going to be the focus of what I'll talk about today. So I'll talk about these two clinical research efforts. The first involves collaborators that I have both at the University of Pittsburgh and our Carnegie Mellon University. And this is a study that I lead. The second is our brain donation program, and this is something that I'm affiliated with. So I'm going to sort of break the talk into these two different sections, and we'll sort of pause after the first if there are any questions at that time. So the first study on motor unit population changes in ALS, like I said, involves both clinicians at the University of Pittsburgh that are collaborators, as well as scientists at Carnegie Mellon University. And I just wanted to highlight the fact that this research trajectory in my lab has been started and initiated by funding support from the ALS Association's seed grant program. So this was funding that was received in 2003 that has led to the start of this pilot study. And that's really what it is that I'm sharing with you today, is a pilot study for us to start to address these aspects of our research. So as a little bit of a background for what I'll talk about, we know in ALS that what happens is that there are problems with the communication between motor neurons, so that's this nice big blue guy here, and its connection with muscles. And so this neuron muscle communication is really what's important in understanding how this functions normally, and then what goes wrong in ALS is obviously a really big area of focus and interest. But I wanted to highlight that one of these motor neurons and its one connection with a muscle is considered a motor unit. So it's one unit of what contributes to the functioning of that particular muscle. And so I bring that up because there are these two major muscle types that are present in all of us. The first is called slow twitch muscle fibers. And so that's this picture here. And so you can think in terms of running, a slow twitch muscle fiber would be involved in your long marathon running. So these muscle fibers are slow to fatigue. So they don't tire out very quickly, allowing you to run that really long marathon. The other type is a fast twitch muscle fiber. And so these muscle fibers are sort of the opposite of slow twitch. So if we go back to the running analogy, this would be like a sprint. So these muscles are powerful, but they very quickly tire out or fatigue. And so I bring these two muscle fiber types up because there have been suggestions, at least in animal models of ALS, that there's a different vulnerability. So a susceptibility fiber type. So specifically in animal models of ALS, fast twitch muscle fibers are thought to be more susceptible to the disease process, whereas the slow twitch fibers seem to be more resilient or resistant. However, the basis for our research study is really that there is little known about the ways that ALS affects these different muscle fiber types in people. And so really what led us to starting this study was my team's experience and our collaborators' experience with looking at individual muscles and individual motor units more specifically in various patient populations. So what I'm showing here is a picture of some previous studies by our team. And this was a person that had suffered a spinal cord injury. And even in the presence of this spinal cord injury, was still able to allow for muscle specific activation so that we could understand potential ways to cure and treat that specific spinal cord injury. And so what I'm showing here is sort of this sticker, that's a brown sticker. And each of the little lighter yellow holes is a place where we are able to tap into and listen to what the muscle is saying. So I've highlighted a couple of different channels here. And each of those channels is able to give us information about what's going on inside that muscle, listening to what the muscle is saying. And that's often called electromyography, or EMG, is how I'll refer to it in the subsequent slides. So again, our team had a lot of preliminary work with EMG-based studies, and this is a high-density EMG system. And we wanted to apply it to determine what the impact of muscle fiber type is in people with ALS. And so ultimately, this is an effort to better understand what is happening in ALS. As you are probably all aware, there are multiple mechanisms that we know contribute to ALS. The big problem is that we don't have a clear-cut understanding of what's a cause and what's an effect. And so what we're trying to do here is, again, take what we know from animal models, that fast twitch muscles, the ones that are powerful but fatigue quickly, those are preferentially vulnerable in animal models, but we don't really know if they play a role in actually human muscles and human ALS. And so we want to understand this muscle fiber specificity and how this compares between people with ALS and people without, and then how it might change in people with ALS over time. And so these studies hadn't been completed until we started this. And we were really trying to determine the role of these different muscle types, better understanding the mechanisms that underlie ALS, and specifically that exchange of vulnerable muscles or vulnerable systems to resilient systems. And so I'll give you a little bit of the basics of our research study. So for this research study, what we do in a visit, which usually takes about 3 hours, is collect information about 4 target muscles. So 2 in the upper arm and 2 in the lower leg. And so these muscles are ones that act in opposition to one another. And what we do is ask participants to do various sustained contractions of that particular target muscle. And what we do is listen to the activity of the muscle. And what I've shown here to the side in the slide is a sensor that we place on top of the muscle of interest. And this is specialized for us to do motor unit analysis. And so a particular research participant would do these sustained contractions of these 4 different muscles. And what we do is ask them to maintain different intensities of that contraction as we apply some pressure. So they're really exerting different pressures with their muscle. And we're trying to induce fatigue. So we're trying to sort of figure out when the fast twitch fibers and then the slow twitch fibers will fatigue, and get a sense with these different held contractions when that occurs in someone with ALS or without ALS. And so just to give you a little sense of what this looks like, here is an example of one of our ALS participants that has been very gracious with their time in participating in the study. And this is what those upper arm muscles and those sort of protocol for that looks like. So here you can see we're targeting the biceps muscle, and underneath this sort of tape is where that EMG sensor is placed. And you can see here, these are the hands of one of our team members, and they're using a specialized device called a dynamometer that measures the pressure that our participant is applying. And we ask them to hold that particular pressure for as long as they can. And so that is for the flexing the biceps muscle, and here is for extension. So in this case, we're looking at the triceps muscle, and you can see hopefully the little indentation that's associated with that EMG sensor here. And this time our team member is holding that dynamometer that allows us to measure the pressure that our research participant is applying sort of below the wrist area here. So our participant is sort of pushing down with their wrist to extend their elbow and really work that triceps muscle. And again, our sensors are picking up all of the muscle activity in the triceps. And then here's what that study looks like for the lower leg. So again, our study team member is holding the dynamometer, and we're asking our research participant to flex their toes up to the sky. And we're using our EMG sensors to pick up the muscle activity from that muscle that's sort of right along the shin bone at the front of the leg. And then for the flexion, we're asking our ALS participant to push down with their toes to flex their foot. And that engages this muscle in the calf. And so you can see right back here where that is. And again, we're asking our participant to push down and then hold a certain level of pressure until they cannot any longer, or for a few minutes, whichever comes first. So to give you a sense, here's what the data looks like. So this is the information we get from that sensor. So this is just a mess, right? This is just a bunch of lines, blue lines just coming up and spiking at different times. And so this is sort of how that data comes to us. And then we use a lot of computer-based software and programming that we've developed within our team to take this complicated mess here and separate it out into what I've highlighted here, 3 different motor units. So I have 3 of them boxed here. And so what this shows is basically, here's 1 motor unit, 2 motor units, and 3 motor units. And anytime we see these black or gray boxes, that indicates that that motor unit was working during that time. So this top one, little bit lazy and contributing to this muscle function here. This one a little bit more involved, and this one involved to the greatest extent. And what we're able to do, as a little reminder, this is just what the motor unit looks like. It's that connection between the motor neuron and the muscle itself, one of those connections. And what we're able to do is use some other analysis metrics to determine, is this particular motor unit a fast twitch motor unit or is it a slow twitch motor unit? And then we can look at how it functions in the course of these studies and if it is undergoing fatigue and if that differs in someone with ALS versus someone without. So here's a little bit of an example of our results. And so you can see that it's differentiated between these top panels for the triceps muscle are the slow twitch muscle fibers. And then these bottom panels are under the high frequency showing the fast twitch muscle fibers. And so if we just take a look and compare the slow twitch fibers in someone with ALS versus someone without ALS, whenever you see the pink or the orange or the red coloring, that's suggesting that there's compromised function. So you can see in the person with ALS, both their slow twitch muscle fibers and their fast twitch muscle fibers have compromised function in many cases compared to a person without ALS, in the blues and the greens, indicating that the muscle is able to adapt to this long-term activation. So we can see that there are some discrepancies between someone with ALS and someone without ALS, and we can break this down, as I said, between the two muscle fiber types. And so to date, the data that I'm showing you is just from one person with ALS and one person that does not have ALS. And so I just highlight that these are very preliminary findings. But what we're seeing so far is that someone that is healthy is only showing signs of fatigue when we're at our highest level of pressure. That MVC is the muscle voluntary contraction, or the maximum voluntary contraction of the muscle. And so it's about 50% of that is when a person without ALS is experiencing fatigue, while as someone with ALS is experiencing fatigue at all levels for 3 of the 4 muscles that we looked at. And so it may be that this observed fatigue and the frequency of it could help us to start to distinguish ALS from healthy individuals. And our additional EMG markers could be tools to allow us to detect differences between our fast twitch muscles, and then potentially changes that occur with the progression of ALS. And so this sort of leads into what we want to discuss as or highlight as our potential impacts. So for example, if we see the same things in people with ALS as we do in the animal models of ALS, which means that the slow twitch fibers are resistant to degeneration, while our fast twitch fibers are more vulnerable and are lost earlier, what we can do for people with ALS is actually develop physical and exercise-based therapies that help to maintain that slow twitch muscle fiber function. And hopefully this would ultimately slow the disease process. In addition, as I mentioned with some of the signatures that we're collecting during this study, we're hoping that we can identify some functional changes that happen, which may be useful to help and aid in the diagnostic process. And these could also be helpful to evaluate specific therapeutic interventions that are being tested to determine if they actually are preventing the loss of these particular motor units. And again, ultimately working to try to not only understand these mechanisms, but to put into place treatments that'll slow and ultimately stop the loss of this muscle function. So even if it is related to fast twitch fibers being vulnerable and slow twitch fibers being resistant, if we really understand what helps to make a muscle resistant, the idea would be to use that to promote resistance across all muscle fiber types. And so what I wanted to just include here, just because I often get these types of questions, and I often hear frustration from people in our ALS community that say, well, I want to participate in research, but for some reason I can't, or, I've had ALS too long, or there's specific criteria that I can't contribute. What I wanted to highlight is for this study, for those with ALS, an ALS diagnosis is enough to participate. So we have two separate components of this study overall. The first one just involves that 1 3-hour visit that kind of goes through those 4 different muscles and testing each of those muscles, allowing that one-time comparison between what's happening in ALS versus someone that is a control, to hopefully compare that in an age and sex matched manner. The second part of the study we're doing, again, to address that longitudinal aspect of ALS to see if we can highlight things that may change over time. There is an opportunity to participate in multiple visits. And so each of these visits would be spread out every 2 to 4 months to determine if there are changes that we can assess in that time, and coordinate and correlate that with disease progression, again, to identify those markers that may highlight what is happening at the muscle level before we see some effects in the person's functional abilities. For healthy volunteers, we're also hoping to include those as comparisons. So really the only participation criteria is that there's no history of neurological illness. And then for our study participants, because we are using EMG studies or electromyography, the only other thing is that there can't be any implanted electrical medical devices. So I'd like to think that our criteria are allowing it to be as open to as many people as may be interested as possible. So with that, I will just kind of pause with an overview from that study and allow time for some questions if there are any related to this work.
Okay. There are a couple questions here that popped through. The first is a clarification with your diagram of the motor unit. They're asking, are you saying that there is only one complete motor unit per muscle? And could you clarify that?
Oh yeah, that's a really great question. So I will go back to this and I'll just go here. So related to the number of motor units per muscle. So there are lots of motor units, individual motor units per muscle. So each of these, so you can see how our neuron comes down and it really branches out to multiple different locations. So each of those is an individual motor unit. So it's the specific muscle component and that portion of the neuron that is controlled by is controlling that particular muscle. So we have tons and tons of motor units, and what we're trying to do in this study is to understand sort of that whole population of motor units. Do we have more fast twitch and slow twitch distribution earlier on, and then we lose maybe only the fast twitch motor units? And so yes, there are a lot of motor units for each of our muscles. And so there's lots of things to pick up. So from this graph that we have back here, this is, really just looking at 3 different motor units, and you can see how much activity is there. But we can record from multiple locations and get tens and hundreds of motor units per muscle.
Okay, great. Our next question, can a person with bulbar also participate in the study, even if the muscle weakness is in the bulbar region?
Yeah, so that's another great question. So for our studies, the site of onset can be at any location. Again, our team has experience in using these types of analyses for a variety of techniques and a variety of conditions. So including stroke and spinal cord injury, and we're able to successfully, listen to basically what your muscles are saying, regardless of your site of onset of ALS.
Wonderful. Okay. So the next couple questions have to do with participating. So I'm going to try to break those up. I guess to start, if somebody was interested in participating in the study, how do they go about that? Is it open right now?
Yes, so our study is open and we are recruiting. The easiest thing would be to just send me an email directly, and I have that shared at the end of the slide deck, but I can also provide it now if anyone would like it. And Ryan, if you want to, add it to the chat box, you're more than welcome to. My
Yep.
email address is C as in cat, L as in love, K as in kick, 39@pitt.edu. So that would be the easiest way, is just to contact me directly. Again, this is a study that I'm leading as part of our team. And so we can, discuss options from there if you're interested.
Cool. And that leads to another question. Is the study currently only being conducted in Pennsylvania? And that you had mentioned it was a pilot program. If it is only available in Pennsylvania, are there plans to expand it to more locations?
Yeah, so thank you for that question. Yes, right now we are basing it out of our research facility at the University of Pittsburgh. And it is a pilot study, so it's meant to be sort of this 1 year to get as much data as we can to determine if, our hypothesis is looking like it might be true, or if there are some interesting things we could learn from this study. Our plan is certainly to, once this pilot project is done, to kind of assemble the data, determine if, what the most interesting directions are to pursue, and then to submit an additional application for funding for a longer term study that would also involve additional locations. The one thing I will add is we do have, and should we expand, we have at our location at least, because of some difficulties, associated with like scheduling our facilities and making it as available as we'd like to our potential research participants. Um, our equipment and the things that we need are pretty transportable, so we actually can make house calls, so to speak, for this study. So that's another added bonus of what we're doing. And so we also try to do that. So again, we are mostly with it around Pittsburgh, but we are open to traveling some distance to get to somebody if they're interested in participating.
Okay. Um, and another more practical question, if participating in the study, is there any program for transportation to be provided, or would the participant be responsible for that?
So we directly with our study don't have a partner for allowing that, but we can certainly, for one of our ALS participants, we did work with them to coordinate with our local ALS association, actually, to help them get transportation to our facility. The other participants we've had so far have had sort of a ride or a plan for that, so they didn't need that additional transportation. We do cover any parking and things like that related to the study visit for those that do participate.
Okay. And then this one's more of a general question, and I'm not sure if you're able to answer it or not, but I don't want to overlook it. And the question is just, are you aware how much research is being done to find a cause and a cure of ALS? And I can speak on that in the sense that there is a lot of research being conducted on all sorts of aspects of ALS, specifically, what's kind of causing it. But if you want to add anything as well. Um,
Yeah, I mean, I would definitely add to that, Ryan. I agree with what you said, and just the the amount of work that's being done and the diversity of the work, I think, continues to be more and more impressive to me. So from, just as a little bit of context, when I first started as a graduate student in ALS, we really focused so much on one animal model when studying that. We were really focused on a specific population, only one population of motor neurons, which are located in the spinal cord. And there were some other limitations of what we were doing, and it wasn't because people weren't interested in other things. There were some limitations just based on what we thought about ALS at the time. Um, but over these past almost 2 decades, we have so many more known genetic links, which have led to so many more modeling capabilities, not just in animals, but in cells derived from humans that donate like skin biopsies, for example, and allow us to look at a disease specific change. And there's so many technological developments in terms of the science and applying that in very creative and strategic ways to try to get at the causes of ALS. And I would also say that it's more and more recognized that the nervous system itself and to control any one muscle doesn't just involve that one neuron. There's certainly value in looking at those individual motor units that I just talked about, but understanding that neuron is part of a complex and concerted system of neurons that are working together, that in some ways may be overwhelming, but it's important for us to try to understand that because these conditions like ALS and other neurodegenerative diseases are complex. And so we have to recognize that and start teasing that apart. So, as a short answer to your question, I think there are so many efforts to do that to really understand ALS. We have so many more genetic links that we've just identified that can take us in different directions and help us to tease apart all of these mechanisms that people think play a role. We can start to get to cause and effect relationships and understand, well, if we fix this, will that help someone with ALS? And that's our ultimate question is understanding things that are causative so that we can target them and really give better options than what we have for people with ALS in our community.
Great, great. And looks like for now this is the last question. And of course, Christy is going to discuss one other study. So if you have more questions about this study, you keep sending them and we can answer them at the end as well. But this last question is more of a practical one. When it comes to participating in the research, uh, pretty much how flexible are you with working with the individual schedule? Would it be possible to align, if a person participated, align it align the participation with a clinic visit?
Yeah, so another really, really great question. So we do try to be as flexible as we can. One of the reasons that we sort of revised some of our approaches to allowing potentially us making house calls was to, help with that flexibility if someone was interested and wanted that. As far as coordinating with a clinic visit, that's certainly possible. However, my only caveat to that would be that, sometimes those visits to the clinic are pretty long and can be tiring, and we wouldn't want to try to tack on an additional 3-hour research study to that. If the participant really wants to do that, we're open to it. But we just want to be very cognizant of the demands that would involve. As far as scheduling, we would try to work with, again, that person's schedule and then trying to make it like, if they have clinic visits scheduled, we could see what works to make it, a date close to, or maybe exactly in between clinic visits, whatever that person might prefer for, cadence or coming to visit us in our lab spaces. So
Great.
we certainly are open to being flexible and working with anyone who is interested.
Excellent, excellent. So I'm going to hand it back to you to move on. If I didn't get to any questions, like I said, at the end, she's got another study that she wants to discuss, but at the end we can revisit them and answer them as well. But I'm going to hand it back to you to discuss the next study you wanted to go over.
Okay, thanks so much, Ryan. So I wanted to just highlight the next two and very related opportunities. Again, just because of my involvement with the ALS community, this is one of the questions that I'm often asked. And I've gotten, actually a couple of emails in the past 6 months asking me specifically about, any program that I know of. And so I really just wanted to share this information with everybody to give everyone a sense of this option that's out there. And these are our brain donation programs. These are in the division that I'm a part of in the Department of Pathology, the Division of Neuropathology. And I'll just kind of review, we have a neurodegenerative brain bank and then a complementary and very much related national sports brain bank. So I want to kind of give you a little bit of the details of this. And if there are any questions, certainly I can take those after this brief discussion. So, it's in terms of neurodegenerative diseases, and one of the questions that was asked is, what are we doing to understand ALS better? Well, one of the challenges is that the brain is so crucial to everything that we do and everything that we are in so many respects that it's not something that we can really access during life. So if you have, a lump somewhere, let's say it's on your liver, those can usually be biopsied. And so if it's, something that's related to cancer, the physicians can then develop a treatment plan that's specific and catered to that particular thing. And with the brain, we're not really able to do that. So that makes it what I call this black box. And so we know some things about it, but there's still a lot about the brain that we don't know. And imaging technologies, and certainly these are significant efforts at the University of Pittsburgh, and as part of my research team, incorporating developing advanced imaging techniques to try to bridge this black box of the brain so that we can maybe make connections between diseases and what we're seeing. But at this point, those imaging techniques can't get us down to the level of an individual neuron, for example, or a few neurons that we know might be affected by ALS. And so, these conditions can only really be definitively diagnosed at autopsy. And at this point, it's really about trying to understand and evaluate the disease process, how much damage that process has caused, and then really trying to connect it back to what that person experienced. So we can, again, really try to tease apart and get to the mechanisms that are associated, that are causing ALS and other related diseases to get to better treatments and ultimately cures. And so I've included this link here, and I can certainly share this, but there's more information certainly that you can find on this if you're interested. But I also wanted to highlight again a related effort to this neurodegenerative brain bank, which is a more recent effort, the National Sports Brain Bank program at the University of Pittsburgh. So this actually is a longer term observational study. And so what that means is that anyone who was involved in contact sports, so hockey, even soccer, football, you name it, any contact sport, if you were involved at any point in your life, there's an opportunity to contribute in this long-term observational study and potentially, and also the brain donation program if that's of interest. But really, this is trying to get at the connection between people or athletes who participated in contact sports, their incidence of, concussions or mild traumatic brain events that may have happened during that time that they were playing. And this is not necessarily for someone who played at a professional level. It's just if you played contact sports at some point in your life. So one of the reasons for this new effort is because of the increased risk of cognitive and psychiatric syndromes that happen and seemingly as a result of exposure to these concussions and traumatic brain events that occur later in life. These have also been linked to a neurodegenerative disease that you may have heard of, chronic traumatic encephalopathy, or CTE. But CTE is also linked to other diseases, including ALS. And so this effort has really capitalized in some ways on the public awareness that has increased over the past few years, in many cases because we've had relatively high profile athletes be diagnosed with CTE. The problem is that there's really a large degree of uncertainty that remains regarding the benefits versus the risks of contact sports. And we really want to try to, clear up this uncertainty because really it relates to the fact that we don't understand the disease, and it's a very limited understanding in what we do know. And so the goals of this national sports bank are to increase our knowledge about CTE and related conditions, which includes ALS. And the effort is involving collecting information about symptoms that are people that directly have the symptoms, but also from their family members. So family members notice, for example, behavioral changes. To get a better sense of the frequency that these conditions occur and how they may relate to one another. So again, CTE and ALS, are they really linked to one another? And this is an opportunity for people to participate again in a long-term study. And the information and data that's collected will be de-identified, which just means it will be anonymous. It won't be ever linked back to any one person. And all of this information will be shared widely with the research community. So not just at our university, but national and international collaborations to try to understand risk factors, but then also things that might impart resilience or resistance to these diseases. And so this kind of folds back into what I talked about initially with that first study, is really understanding resistance and vulnerability. And if we can really see and understand what makes a person or a cell, for example, resistant to the disease process, we can hopefully make that a more widespread resistance that protects that person from these associated consequences. And so I just wanted to finish by saying, from my time in the ALS community, I feel like only the very best people are affected by ALS, and I know that they are all incredibly altruistic. And so I just want to highlight that the decision to make a brain donation is certainly that ultimate altruistic gift that is a hope to help future generations. And like I said, I really just shared these opportunities because I often get asked about this type of opportunity to help. But I also wanted to outline that the National Sports Bank is something that is ongoing. So if you have participated in contact sports, you can start offering data now and information related to that now to try to make those connections. This process is done at no cost to the family, and the family does receive a complete report and the opportunity to discuss these findings with a trained neuropathologist that can, kind of provide more information related to a particular diagnosis. As I said, all of this information is anonymous and it's shared freely with the research community to facilitate what we all hopefully achieve sooner rather than later, which are effective therapies and cures for these conditions. And really, these donations can have impacts on multiple research studies. So I've just included some additional information there if anyone would happen to be interested in learning more about these programs. And so with that, I'd just like to acknowledge the members of my team. The people that are listed in or highlighted in yellow are the ones specifically involved in our muscle fiber type specific studies, trying to understand that difference between fast and slow twitch fibers and ALS. I also wanted to highlight our funding. I mentioned that project is funded by the ALS Association, and our team member Maya, who was in those images with a dynamometer, also had additional funding this summer. And of course, a huge shout out to all of our wonderful research participants. We couldn't do it without them. Again, I just wanted to thank everyone for joining, for the questions already and hopefully to come. And I've included my email address there, and a link to my team's very new Instagram page, so you can follow some of the things we're doing. We recently completed a pit-focused ice bucket challenge to mark the 10th anniversary of that phenomenon. And again, Ryan, I'd be happy to take any questions that maybe have come into the chat.
Certainly, just a few, and you may have covered this, so forgive me. For the brain bank studies, is there an area restriction for those? Are there rules as far as how far a person can be to participate if they so wish?
No, so there are no limits on where someone needs to be. There are some on the websites. There are some additional forms and things that'll give a little bit more information. But especially for the sports brain bank, that's definitely a national research effort. And so it's really no limitations regarding location for those studies.
And going off of the sports brain bank, have there been any results or findings that have been published as a result of that study yet?
Yeah, so this is a still pretty early on.
Mhm.
We don't have any study that has yet been reported, but this was just a very recent initiation that or initiative, I'm sorry, that we sort of undertook, and it's been an effort that we are wanting to promote again because of that aspect of getting information from people in a longitudinal manner.
Okay. Okay. And then just a few general questions, some of them from prior. With the sports brain bank, you talked about trying to find that connection. These questions have to do with have you, in your research, understanding, of course, that your research isn't necessarily focused on these certain connections, but have you noticed a connection between ALS and firefighters? And then there was a follow-up question, whether there was a connection between diseases like Lyme disease and ALS, and if you've heard of any of that in your research and
Yeah, actually, I have heard examples of both of those, examples of people who were, had Lyme disease or another or other viral illnesses, and then felt like that was the start of their ALS. And then the firefighter exposure is very similar, in some ways at least, to what we know is this greater incidence in people who have served in the military. So these environmental exposures is maybe the broad category that would put them under, are really something, and I think in a lot of ways, opens up a lot of questions for us as researchers. One of the people I think that's doing some of the best work related to these different environmental exposures or maybe a particular treatment or vitamin that people have found that is helpful. His name is Rick Bedlack, and he has a specific website that's called ALS Untangled. And if you just search that, if you have anything that you think is a connection, it's a really great. He does a really great job of taking those in. He has a team of people that kind of go through the literature to see, what do we know about this particular exposure? And then if there's, some suggestive evidence, they try to do a study to prove one way or another whether there is a connection there. So I know that wasn't maybe a direct answer to the question, but I've definitely had multiple families who their person with ALS was a firefighter, and I know of a couple who specifically mentioned Lyme disease. But again, that's not largely the focus of my work, but I do think that there are opportunities to offer those up, and things that will hopefully have answers to, as we better understand the disease process itself.
Absolutely. A couple more questions here. I know we're getting close on time. With technology advancing and AI becoming bigger, have you noticed AI being involved in research more or at all?
Yes, actually. And that's, and I'll give you a couple of examples of how artificial intelligence is being used. So, I mentioned, wanting to, imaging and advanced imaging methods being a way to sort of get into that black box of the brain, while it's still functioning and maybe being exposed to certain disease processes. So there are some artificial intelligence um, components to our imaging analysis that are helping to bring out things that maybe we would have missed otherwise. So that's one opportunity or one example of how AI is being used. In others, we have done similar types of profiling of, say we identify something that, in our studies that we think is contributing to ALS. One way is using AI to kind of give us this, distill down what's been done already, or what kind of things do we know about this particular gene or protein, and to help us to, really frame some of the studies and some of the work that we might do from there, to get a clearer picture of how it might be impacting ALS. So those are just two examples, but there's certainly more. I'll give one final one. I am part of a collaborative project to identify small molecule compounds that could be used to stabilize a protein that has a mutation in ALS. And so the idea would be, if we use this small molecule and we stabilize the protein, we can basically take it from misbehaving to behaving properly. And what we hope is that once it's behaving properly, it will, prevent the downstream causes and phenotypes that we see with ALS, or the manifestations of ALS. And so to identify these, we're actually using various software programs that rely on artificial intelligence to profile potential already available molecules out there, to find ones that would stabilize, that would bind to this particular protein in this specific way at this specific location. So we are able to profile like over 4 billion compounds, for example, in a matter of, minutes, in essence, to get to some of the ones that are the best candidates for us to then test further. So it really is streamlining our process and hopefully accelerating the time from, hey, we think we have something here, to we have something that we can test further that can be then moved to the people that we want to get them to.
Okay. And the last question, just because we're running out of time here, and again, this is more of a general question. Just it is asking if you're hearing anything about new meds or treatment strategies coming out soon for sporadic ALS, um, due to trials taking so long. Is there anything on the horizon that you're aware of, being a researcher, that might be coming out?
Yeah, so there's nothing in particular that comes to mind that's right there. Most of those trials, I would say that we are doing our best to, be very intelligent about the trials that we're designing to, make sure that we are getting to an answer of whether it works or not more quickly. And while that's sort of a aspect of the research that I'm less involved with in terms of the clinical side. As a basic researcher, I do believe that we are, understanding more and more every day about the mechanisms and the potential things that we could target, that could lead to better for people in the way of treatments that are affected by ALS.
Okay. Well, we are out of time, but those were fantastic questions and amazing answers. If we did not answer your question or you think of a question afterwards, please feel free to reach out to myself and I can try to get you an answer from Dr. Kolarcik. I know Dr. Kolarcik also put her email address up there if you are interested in any of the studies that were discussed or you would like more information or how to participate. It sounds like you can reach out to her directly. We just want to thank Dr. Kolarcik for the wealth of information about the amazing research being conducted. We thank everyone in attendance today. Again, we hope you found the information helpful. Just a reminder that you will receive a short survey when we end this webinar. The survey should also be included in a follow-up email that will be sent out to everyone in attendance tomorrow. So again, thank you, Dr. Kolarcik, and thank you to everyone in attendance, and we hope you have a good rest of your day.
Thank you, everyone.